Sorry, everyone who's just been listening into us preparing, but I just wanted to welcome everyone to this month's cancer theme webinar. There's a couple of housekeeping things. Obviously, just if you could just double check that you are on mute, that would be great. And the format we'd like to go with today is it perhaps have the speakers present and then ask questions all together at the end. So if you could pop your questions in the chat, which I'm sure everyone's getting more and more used to as we've moved over to Zoom recently, then I can either read them out or I can unmute you at the end if you want to ask them yourselves. That would be fantastic. So our first speaker today is Dr. Stephen Pacey. He's the Preclinical Facilities Manager at the Wales Research and Diagnostic PET Imaging Centre. And today, Stephen's kindly agreed to provide an overview of the exciting cancer imaging capabilities that we have here at Cardiff University, and to help guide us towards how we might apply these sophisticated imaging technologies for our own research interests. So over to you, Steve, if you want to share your screen and tell us all about what PET imaging can do for us.
Just setting that up, right. Okay. Can everyone see that? Has that come through now?
Yeah, it's just fading in now, Steve.
Brilliant. Okay. So yes, thank you very much for the invite, Helen. And it's delighted to have the opportunity to tell you about the various things that we've been developing at PETIC and what these how these imaging technologies are available and how they might help you. So if I start off with just a brief introduction to PET imaging itself. So a PET scanner simply detects injected radioactivity. So that means we need to inject our subject with a small amount of radioactivity before any scan. And then we usually wait an appropriate uptake time before we image. And the scanner can detect, it gives you a 3D distribution map of that tracer. So what that actually means is that the biological relevance of the scan is determined entirely by the pharmacokinetics of the tracer. So it has very little to do with the setup of the scanner itself, and it's all to do with what you've injected. So this is a PET scan. So what's special about a PET scanner is it's incredibly sensitive. So it can detect just picomolar concentrations of radio tracer. And the other thing that's interesting about it is that it's quantifiable, so you can compare one scan to the next. So we get a 3D tracer distribution map, and the preclinical scanner has a voxel size of 400 micrometers, which is not a brilliant resolution as imaging goes. But on its own, a PET scan does look something like fuzzy blobs in space. So you couple it with an anatomical imaging methodology such as CT. And when we put the CT on, hopefully you can see that this is a mouse. And the bright spots are lymph nodes, lymph metastases and lung metastases in a mouse. And this is a mouse that's been injected with FDG. Of course, we have to get the radioactivity from somewhere. So we have a cyclotron in the basement of the hospital, which is a miniature particle accelerator, and that generates a whole host of radioactive isotopes. But on their own, these isotopes don't really have any biological relevance. So you need to do some chemistry in situ to be able to attach those isotopes onto biologically relevant molecules, and then you have a tracer. So we have a GMP, which is good manufacturing practice, clean room, where we can make radio tracers to clinical standard, and we run the routine cancer diagnostic service for Wales. And we produce the tracer in-house. And then we have quality control labs that prove that we've made what we think we've made before we release it to the clinic for the imaging. We also have, alongside that, equivalent sized radiochemistry research labs where we can do tracer development for research purposes. And we have a research scanner, which is a small animal PET CT scanner, and we can scan rats and mice. And it's kind of big enough to image guinea pigs and rabbits, but we don't have the holding space set up for that. So the scanner itself is housed in its own dedicated holding facility, and we've got a radioactive area and we've got other holding areas. And so it's been deliberately, purposefully set up so that animals can be brought in from any of the facilities in the university and brought in and housed long term for the length of time of the study. And then we can also bring animals in from outside of Cardiff University as well. I was supposed to change slides. There you go. So having explained that it's all about the tracers as to what the scan shows, then we need to look at what tracers are available. So the standard tracer is FDG, which is radioactive sugar. And we use this for our clinical service, and we image the patients, and the sugar is taken up very strongly by the tumors, as you can see again in this same mouse here. But because it's simply glucose, it's also taken up by other cellular processes as well, just by ordinary cells. And it's quite strongly taken up by the heart. So I don't know, hopefully this comes through as a movie. And you can see you can use the FDG to get an image, like a cine movie of the heart beating. This is just a healthy mouse to prove the technology. But then the scanner is detecting the emission from this fluorine atom that I've labeled in red. But if you were to take that fluorine atom and stick it onto a different molecule, you get a completely different scan. So this tracer now is the second tracer that we brought online, and this is F-DOPA, which highlights the dopamine signaling pathways in the brain. And here's an image of a rat, and you can see that it's got uptake on the left hemisphere, but the right hemisphere, there's no uptake whatsoever. And that's because this rat has been lesioned to have Parkinson's-like symptoms on that side of the brain. And just to show the sort of subtlety of the chemistry, if we take the fluorine, the same fluorine, but we stick it onto a different molecule, falapride, if you look closely, you see actually a lot of the central motif of this molecule is the same. However, it's not quite the same. And this binds to the D2 receptors and shows postsynaptic binding. And so this is actually the same rat scanned a week later. And now you can see that the D2 receptors have not been damaged by the lesion, and you get uptake now. So this is just a slide to show just how subtle the effects of the chemistry can be and how you can manipulate the scans by the chemistry. Move to the next slide. Sorry, it skipped. There we go. So alongside those tracers that I've already shown you, which we make to GMP standard, which means we can use them in patients and in the research, we also make 18F PSMA, which is a prostate cancer tracer. And we make flumazenil, which is another neurotracer. This is looks at GABA in the brain. And then for research only, we can make, at the moment, we can make TSPO or DPA714, which is a neuroinflammation marker. It binds to the TSPO protein. And a tracer that we're looking at translating into our facility is SV2A, which is a neuronal density tracer. So these are the tracers that we have readily to hand or shortly are going to develop. But we also have a very talented organofluorine chemist, Dr. Matt Treadwell, whose research is all about finding new ways to put fluorine on interesting molecules. So if you have your own specific marker that you'd be really interested in looking at, we could certainly have a conversation about whether or not that's a molecule that would be easily labeled or not, and whether it's something that we could go forward with to develop. But the difficulty of the process is determined entirely by the chemistry, and some molecules are much easier to label than others. And Matt's the guy who would tell you what's a good idea and what's not, or what's a very challenging chemistry project and what's a simple one. But moving on from the fluorine chemistry, my research is actually focused on trying to put Matt out of a job, really, because what would be nice is if there was a way that we could shortcut all of that chemistry development that's required to develop a new tracer. And one of the ways in which it would be nice to do this is to harness the power of biology and the control that biologists have on different molecules and all the advanced therapeutic medicinal products that are available. So it would be really nice if you could take antibodies or cells or other advanced therapeutic products and directly label them. And the problem is one of scale. So if you look at an antibody, the glucose is just a small side chain on the side of an amino acid of that antibody, and the antibody itself is just a small part on the side of a cell. And so the circulation times of these molecules, or these biomacromolecules and agents, is very much bigger, much slower than the small organic molecules. And so you would image these days after injection rather than simply minutes or hours after injection. So fluorine, with its 2-hour half-life, is ideally suited to the organic molecules, but is no good really for imaging these biomacromolecules. So at PETIC, we've developed the technology. Sorry, it's a bit slow, this. We've developed the technology to produce a new isotope, zirconium-89, and this has got a 3¼ day half-life, and that allows us to image for up to 17 days from a single injection. So now I can directly label all these biomacromolecules and look at them as they circulate around the body. So the first applications for zirconium were based around antibodies and a simple chelation chemistry reaction. So we can take this desferrioxamine, we can bind it to the amine side chains of an antibody, and then we can take that conjugated antibody and stir it up with some zirconium, and we'll turn it into a PET tracer. So this chemistry is the same no matter what the antibody targets, whereas for the organofluorine chemistry, you have to invent the chemistry each time specific to the molecule you want to label. So we've done some work imaging, synthesizing antibodies, and here's an example of an anti-C3d complement antibody. This is a neuro application. So these mice were meant to develop multiple sclerosis, and we were hoping to image the immune response in the brain with that. However, the mice didn't, in this case, get ill. However, this one mouse did get an eye infection, and you can see quite clearly you've got this binding, this localization of the tracer to the eye region of this one mouse to show that the antibody was working. And just to show the variability or the flexibility of this approach, it doesn't have to be full-size antibodies that we label. So when I'm scanning with zirconium, I can scan 3 mice at a time. So each 3-mouse tile is a single scan. And you can see you've got the time after injection across the top. So 0, 3, up to 144 hours. And so the top row is an antibody fragment, which is about 40 kilodaltons in size. And you can see that that starts to accumulate in the kidneys, and actually that's quite typical of molecules of that size. They get trapped in the kidney tubules. Then we radiolabeled a small peptide, and this was extremely rapidly excreted through the bowels. And then just for comparison at the bottom, you've got the free zirconium, which if it does leach out of the labeled tracer, what you find is that you get that it localizes into the joints and the spinal column. So this is just the free zirconium. So that's kind of protein labeling, but we can also look at cell labeling with this antibody as well, with this isotope as well. So we can take cells that are due to be injected, and we can pre-label them, we can implant them, and this allows us to image cell distribution for up to 17 days post-injection. So here's an example of a project that we did with Anasia, and this is still an ongoing project. And we took T cells and we labeled them, and we injected them through the tail vein into a mouse. And you can see that you've got some signal as the activity is homing to the tumor. And this is 5 days post-injection. And then we were able to follow that right up to 17 days post-injection, you can still see some signal. And you can see this is the tumor here. There's a little bit of residual signal here. And what's really nice about this study is that the tumor was actually kind of eaten away by the T cells as well. So it does seem to suggest that the cells retained their activity after labeling. We can also use this technology to radio label exosomes, and we've been looking at labeling those and imaging those. And so in a project with Florian Siebsenrubel and Alid Clayton, we took some radio labeled exosomes and we looked at the effective injection route. So the 4 tiles on the left-hand side, you can see are images of mice that have been injected with exosome that was injected into the intraperitoneal cavity. And then the 4 tiles on the right-hand side are the similar wild-type mice injected with the same exosome, but this time injected via the tail vein. And you can see that they've got a vastly different distribution. So this is some of the sorts of things you can do with PET imaging. So this was 40 hours post-injection. So that's a sort of a whistle-stop tour through some of the things that can be done with zirconium. So it's very good, in summary, at allowing you to track things that you've injected that you hope are going to home to a site of action. But there are also, there's different applications where you might want to inject things and get them to stay put. And the biologists working on these applications tend to be looking at a longer time scale than just this 15 or 17 days. So vanadium-48 has got a 16-day half-life, and this would allow us to image potentially for 2 months after a single injection. So the sorts of things applications I've got in mind for that are looking at grafted cells that are grafted for repair, and you're hoping that they stay put at the site of injury, or even scaffolds themselves that you might label and implant and watch how they slowly get eaten away by the cells, by the repair processes, or cells seeded on scaffolds, or things like looking at agent release from bone cements and things like that. So vanadium-48 is at an earlier stage of development. We have a PhD student working on the chemistry, and we're trying to get good purifications and develop the first labeling strategies to then be able to apply it to biological systems. Another thing that you can do if you want to look at even longer time scales, and this is something that I'm investigating setting up, shall I say, in the PET Center, is if you engineer your cells to have a function that normal cells don't have and implant those cells, then you inject a radio tracer that is engineered, that is designed to hit the target that the cells have been engineered to have. You can then use a short-lived tracer, an image, and of course you can keep re-injecting that tracer many times, and you can wait months or years. You're not limited by the half-life anymore of the pre-labeled cells, because of course they've been engineered to permanently express a function. So one example of that, which is of interest, is thymidine kinase. So thymidine itself as a molecule is actively shuttled across cell membranes, and it's phosphorylated inside the cells by thymidine kinase. And once it's phosphorylated, it can't come back out of the cells, and it goes on to be written into the DNA. If you slightly modify this molecule, so here we put an iodine on instead of a methyl group here, and a fluorine in the backbone so that we can see it, this FIAU is actually not recognized by the thymidine kinase, the mammalian thymidine kinase, but it is recognized by the herpes simplex virus thymidine kinase. So if you engineer cells to express the herpes simplex virus thymidine kinase, they will uniquely phosphorylate the FIAU when your host cells won't. So here's an example from the literature. So this is a tumor that's been engineered to express the herpes simplex virus thymidine kinase, and here's a tumor which has not been engineered to express it, and you can see a marked difference. There's also uptake in the stomach, and this is the excretion through the bladder. So this is just one of many reporter genes that are available, potentially available. And I've got an interest in developing this, but at the moment we don't have it on tap in the facility. Another future work where we're sort of at the stage of writing grant funding and hoping to be able to develop this is PNA constructs for in vivo imaging. So PNA is a peptide nucleic acid, and it's kind of resistant to RNases and DNases, and it allows you to make antisense matches to mRNA, and it's used quite a lot in silencing of mRNA expression, but it can also be used for imaging. So if you take an a PNA sequence designed to be antisense to an mRNA of interest, that can bind to the thing, to the mRNA, and be trapped inside the cells. And to be able to see it, you need a signal of some sort, either a fluorescence or a PET or whatever. And you also need some way of getting it into the cells. So you've got a receptor targeting section. So these chimeras are something, again, that's in the literature. And here's an example of one which has been made, but this time in the literature, it's a SPECT agent, but we think it would be good with zirconium to make it a PET agent. And this is the example of it, where the PNA sequence has been designed to target mutant KRAS sequence in pancreatic cancer. So we're looking at whether we can develop this for use in Cardiff with collaborators from Bath who have a peptide synthesizer. So we're in the process of trying to generate funding to bring this kind of technology online as well. So that was a whistle-stop tour through as much as I could show you within 20 minutes. And I'd be happy to, well, first of all, I'd like to thank all these many people from all different fields who've helped develop these technologies. And I'd be happy to take any questions at the end of the session or talk to people afterwards. Thank you very much for your attention.
Lovely. Thanks, Steve, for that. That was fantastic. Just loving all those images. They're just amazing. Obviously, we've all got lots of questions, but as we mentioned at the start, just in case anyone joined later, we're going to take questions for Steve at the end after Anne's talk. Anne, do you want to start sharing your screen?
Yes. Yeah, here we go. Right. Okay.
But just to introduce Anne first up. So we've got Anne Measure from the Division of Infection and Immunity, and Anne's research into T-cell trafficking has been hugely instrumental in identifying novel approaches to boost immunity and to protect our bodies against the viral infections and cancers, and also things like neurodegeneration associated with Alzheimer's, by increasing the T-cell homing to the diseased tissues. And Anne's research has made significant advances in the field, and some of these recent findings have revealed an essential role for L-selectin in the recruitment of killer T cells to the flu-infected lungs for virus clearance. And she's also been able to show that enhancing the expression of L-selectin on cancer-killing T lymphocytes can increase their efficacy in adoptive T cell cancer therapies. So just hugely amazing findings. And I'm really looking forward to hearing Anne's talk today about her latest exciting research into imaging T cell trafficking in cancer immunotherapy.
Well, thank you very much, Helen, for that very flattering introduction. I hope the next 20 or so minutes, I'm actually going to give a little bit of biology background of why we think it's important to image T-cell trafficking in cancer immunotherapy. I'm sure many of you are familiar with some of this, but please bear with me. So we've known now since, the pioneering studies of Jérôme Gallon in France a long time ago now, 2006, I think the science paper was published, that the composition of the T cell infiltrate of they started working on colorectal cancer, but this idea that the precise makeup of the T cells inside the tumor microenvironment actually correlates with patient outcome. And you can see that from this is a bit of a noddy diagram, but basically many TILs, tumor infiltrating lymphocytes, patients do better than those with fewer TILs. And a lot of work is going into this analysis, and it's being developed and used as a sort of immunoscore. So the types of CD8 killer T cells and whether they synthesize expressing interferon gamma, whether they have the TBET transcription factor, which defines their lineage inside or at the invasive margin. And that's just quite an old review now. But I mean, I think this has revolutionized imaging in cancers, in the sense that now we have new technologies of doing multiple colors, as it were, or multiple antigens within cancer tissue, so we can actually generate this data about immunoscore. So I'm not going to talk about that, but really what interests us is if there is a relationship between killer T cells and patient outcome, then two questions: where do they come from? And why don't they work inside cancers? Because obviously what we're looking at here is resected cancers from cancer patients. So, you know, and we know obviously that the tumor microenvironment is highly immunosuppressive. But our argument is really that T cells need to get there, need to home there. So again, many of you will be familiar of this approach that's been pioneered by Steve Rosenberg at the National Cancer Institute, again, many years ago, of saying, well, if TILs are inside cancers and they correlate with patient outcome, can you actually increase their numbers and infuse them back into patients, and in order to try and overcome the dominant immunosuppression? So is it simply a numbers game? If you can present more T cells to the tumors, will you switch the I guess the seesaw of immunosuppression versus immunoactivation? So again, many of you are familiar, this has been really very applied to resected melanomas, because obviously it's a skin cancer, so it's easy to be resected. TILs have been isolated and expanded very much in vitro, and then infused back into the bloodstream of patients. And obviously it's not 100% successful, but there are 50% or less than 50% overall response rates, and there are trials in all over the world. So one question that we're interested in is, does killer T cell homing contribute potentially to the failure of T cell therapy? Are the T cells just simply not getting to the tumors in sufficient numbers to outcompete cancer growth? And then we have the complement of this, which is really the remarkable success of CAR T cell therapies to treat blood cancers, leukemias. So this is CD19 CAR T cell therapies. This is where the patient's T cells is basically re-engineered with a cancer-specific or leukemia-specific T cell receptor. In this case, it's the T cell receptor that binds to the B cell lineage marker CD19. And this has, I guess, a remarkable success. And so this has really stimulated research into developing CAR T cell therapies to treat solid cancers. And that's the sort of, the big hurdle that we have to come. But just so that you're aware, up at the Cardiff and Vale Hospital, patients with diffuse large B-cell leukemia are being treated with CAR T cell, CD19 CAR T cell therapy on the NHS Wales budget. So it is happening in the clinic. It was rapidly approved by NICE. And in fact, there are several research-related projects going on to try and look at how fit those patients' T cells are once they've been engineered outside the body and re-infused. And just generally, the role of T cell trafficking in this, I've already said this is success or failure of CAR T cell therapies for either blood or solid cancers is really not well studied. So we've known for a number of years that tumor blood vessels themselves are dysfunctional. I mean, tumors or solid cancers, in order to grow, stimulate angiogenesis. They need obviously a blood supply for nutrients and oxygen, and to remove waste products. But this rapidly growing tumor vasculature doesn't look right, basically, and is dysfunctional. So if we look on the left-hand side here, this would be a cartoon of the vascular bed in a sort of normal or even inflamed tissue. So you've obviously got the major feeding artery, feeding into arterioles, then into capillaries and post-capillary venules, and then collecting veins. So if we look at a high power image of post-capillary venule, and this is the part of the vasculature where T cells are normally recruited into either normal tissues, such as lymphoid organs, or inflamed tissues. And the blood vessels are very well organized. So in green here, we've got the endothelial cells, which form a continuous lining to this blood vessel. Sorry, they sort of seal the tube, as it were. And then we've got this very nice tight wrapping around of pericytes, which are like a fibroblast, a stromal or mural support cell. But in tumors, the vasculature is very disorganized. You don't see this nice organization. In fact, when you look at the tumor blood vessels, you can see endothelial cells, but the most remarkable thing is that the pericytes have basically lost contact with endothelial cells. And the result of that is the blood vessels tend to be very poorly perfused and even leaky. And in many preclinical animal studies have shown that poor T cell recruitment or T cells, particularly CD8 T cell recruitment, is controlled or restricted by multiple immune evasion mechanisms, which I'm not going to go into, but we can talk about at the end, or talk, very happy to talk about it afterwards. So this is just a new study actually that was accepted for publication 2 days ago in Med, which is a new Cell Press medical journal. So this is a very nice collaboration that we had with John Greenwood at UCL. So he had been working on large 1, and antibodies to large 1 he'd shown interfere with TGF beta signaling in vascular endothelium. And what he'd shown is that it was correlated with disorganized blood vessels in actually eye diseases, in diabetic retinopathy, where the pericytes tend to leave the blood vessels in a similar way to I've shown you with tumors. So they had developed a therapeutic antibody which would block this signaling pathway. And to cut a lot of work short, we were involved just in a very small part of it. So the large blockade was shown to normalize tumor blood vessels. So essentially it lined the pericytes up again, and the tumors were much less leaky. The tumor blood vessels were much less leaky, and the tumor microenvironment was much less hypoxic, as it were. So what we did with John is ask, well, does this translate to a more effective adoptive T cell therapy? Do these improved stabilized blood vessels actually end up recruiting more T cells and are more efficacious? So what we've got here is a simple mouse model, which I'll actually introduce in a couple of slides. So we're looking here at the growth of a subcutaneously implanted, very artificial, I'm afraid, but a melanoma expressing a surrogate tumor antigen. And this is growth of the tumor in mice without any T cell transfers. What we've done here in the green line is we're injecting basically T cells, CD8 T cells that have a receptor for this surrogate tumor antigen. So it's a very highly engineered model, but you can see it works. When you transfer these T cells, F5B6 T cells, they actually restrict tumor growth quite effectively, then eventually control is lost. This is with an isotype control antibody. When you combine it with a 15C4 antibody, you can see you get a synergistic combination effect. And in fact, these tumors almost look like they're regressing. And when you're now trying to image T cell trafficking, and this is actually looking at end stage now. So this is collecting the tumors and then doing multicolor immunohistochemistry. And we have a marker for endothelial cells here in blue. We have a marker for donor T cells, so we can distinguish donor T cells that have been injected in in, which is the therapy from the host's own T cells, because obviously the mouse has its own T cells. So without any transfer, you can see obviously a lot of blood vessels and very few infiltrating host T cells in green. With the control antibody, you've got a few more donor T cells coming in. You can see them in red, and obviously the red and green make yellow, the combination of the markers. But you can appreciate when you add this vascular normalizing antibody, you see a massive infiltration of T cells. So we, intuitively you think those T cells, because they have a tumor T cell receptor for a surrogate antigen in the tumor, are actively controlling tumor and leading to this result. So this was rather pleasing. And actually this therapy combines with chemotherapies and has some clinical relevance. So as I say, the paper will be out soon for those of you interested. So this approach of targeting blood vessels is not new. There are clinically approved agents already. You will be familiar with anti-VEGF, VEGF and anti-VEGF receptor. Sorry, that's a typo. That's been approved for colorectal cancer and other cancers. So what we think this is doing is starting off with these anergic tumor blood vessels where we've got leaky vessels and low T cell infiltration. Vessel normalization is gathering the parasites, around the blood vessels again, some reversal of endothelial cell energy and upregulated homing molecule expression in order to recruit T cells. So large one, what I've just showed you, is another way of doing this. Anti-endothelin B receptor has also been shown by George Kukas, sorry, it's Friday afternoon. George Kukas and Lazan, very elegant work showing that endothelin B receptor signaling in endothelium downregulates adhesion pathways. And there's also mechanisms of targeting TNF alpha to directly activate tumor blood vessels. So one step beyond that is really to mature these vessels into what's called professional T cell trafficking vessels, called high endothelial venules, which you normally only find in lymph nodes, because this is their role, to extract T cells from the bloodstream, irrespective or independent of T cell receptor specificity, in order for those T cells to then survey the lymph nodes for peptide MHC complexes for subsequent activation. But some work, well, really it should start with work of Arwin in Cardiff, showed very early on with a depletion of a highly immunosuppressive CD4 Tregs, you get spontaneous HEV neogenesis, which correlates with control of an even regression of tumors in an autochthonous tumor model. And then there are some recent work with dual blockade of PD-1 and VEGF and vascular targeting of light, which signals via sort of lymphotoxin beta receptor, also seems to push the vascular maturation pathway down this HEV route. Now this, and that correlates with increased control of tumor growth or cancer cell killing. And these have been shown to work even in quite refractory mouse models, such as glioblastoma. So, but they are all obviously preclinical models. So time will tell as to whether these will translate into clinically relevant therapies. So just going back to T cell homing, so we can get more T cells in there. I've shown you with blocking anti-large 1. So what are the molecules involved, and can we find out what they are, and can we manipulate them in any way? So this is just a teaching slide. I hope that many of you have come across the multi-step adhesion cascade of how leukocytes are recruited from the bloodstream into tissues. And this is really a sequence, a highly regulated sequence of adhesive interactions, which is controlled in time and space, as it were. So it's very tightly regulated. So the first event is, if you imagine leukocytes are just obviously being carried at very high speed through with blood flow, then the first event is sorry, I'm afraid I'm at my mum's and the family have arrived and the dog started barking, so please bear with me. So the first interaction is very much a transient, reversible interaction mediated by selectins, low affinity receptors for glycans. This is followed by an activation-induced arrest, which is very much dependent on chemokines presented on the inner endothelial surface inside blood vessels, which activate integrins and leads to arrest. And then obviously following that, there's directed transmigration and infiltration of tissues, which is very much chemokine receptor driven. So we started at the beginning, which is the first event, enrolling, because if cells don't roll, they don't make contact with endothelium, therefore they can't get activated. They can't be activated by immobilized chemokines. So we started at the beginning with selectins, and we know the dogma was that L-selectin, which is on naive T cells and a type of memory T cell, this binds to a ligand on these specialized high endothelial venules that they only really normally find in lymph nodes. PNAD it's called, and this is important for getting T cells into lymph nodes for subsequent activation. But in inflamed tissues, then the dogma is L-selectin is downregulated on activated T cells, and so it can't be involved in homing to inflamed tissues, and other selectins are important here. And these are now selectins on the are activated on blood vessels, so E and P selectin with a ligand on activated T cells. So we looked at this dogma again, and essentially we found that it wasn't quite true. And essentially we found that L-selectin is actually expressed on early activated cytotoxic T cells, killer T cells, and is essential for homing to virus-infected tissues such as lungs, influenza-infected lungs. So we sort of asked if this pathway could be important in cancer immunotherapy or in cancer immunity. And just again, quickly summarize, go through the cancer immunity cycle. Again, I'm sure you're very familiar that we start this very elegant teaching slide from Chen and Melman. We start with cancer cells dying and peptides being, cancer antigens or peptides being taken up by dendritic cells and transported to draining lymph nodes, and presented as peptide MHC, and this is where endogenous T cells will be activated. We know L-selectin is absolutely essential for this trafficking into lymph nodes via these specialized HEV. Once T cells are activated and released into the bloodstream, then obviously they will need to locate the cancer. So they need to be able to be recruited from tumor blood vessels. And as I say, the dogma was L-selectin is not important for that pathway. So, however, if you do things like this, which is look at Ruba and Duraj in the lab, it's unpublished data, looked at cell L-selectin or cell, the gene messenger RNA level in the cancer genome atlas, then you do see correlations between high L-selectin and improved patient outcome in melanoma, ovarian cancer, head and neck cancer, but not in these two kidney cancers. So, it suggests that maybe there is some this is worth studying. So we simply did a mouse model, and this was a mouse model of adoptive T cell therapy, and we used 3 different types of T cells. So T cells that have L-selectin, normal L-selectin, T cells don't have any L-selectin at all, and then we've engineered a mouse where the L-selectin is not downregulated. It cannot be silenced or shed, which is what happens in the wild type situation here. The T cells that we're putting in are mostly naive T cells. I'm not going to explain why today, but I can go on to that. And the model is we implant tumor cells subcutaneously, let them establish for 6 days. Then we do a sublethal irradiation, and that simply is to knock out the endogenous immunosuppression, such as Tregs, which is very Treg dependent model this. And then we inject naive T cells on day 7, and we can follow tumor growth because the tumors have luciferase labels, so we can use the bioluminescent imaging. We also measure tumors and then out to humane endpoint. And this is a very T cell dependent model. So it depends on the number of T cells that you inject. So the green line here is no T cell transfers, tumor growth. A yellow line is third party, so T cells that don't have a receptor for cancer antigens, whereas this is increasing number of T cells. So this is quite a nice therapy, you know, proof of principle that the numbers of T cells is important. So when we applied this with our 3 different types of T cells, you can see, I think, T cells going in all have the same level of T cell receptor, but the knockouts don't have any L-selectin, and the LDPs, as we call them, have high L-selectin. At the end of the therapy, there's no L-selectin on the wild type T cells, but it's maintained in our enhanced T cells. And then again, just to highlight that these, in the red line here, these are our enhanced L-selectin T cells, which are better than wild type and better than knockouts, which really don't do anything at all. And this is the individual growth curves. Obviously, it's not perfect, you know, you're not getting absolute tumor regression, but this we know that these will work synergistically with the large 1 blockade, which I've showed you. We also know that they'll work synergistically with anti-PD-1, so with checkpoint blockade inhibitors. So whatever it's doing, it's not doing the same as those targeting agents. And we know at end stage, we see more CD3 T cells in the tumors with the therapy that works, and more CD8 T cells. But of course, we don't know when they got there and when they started to do their job. And that is the problem, or the difficulty with cancer immunotherapy. The other thing we know, actually, which was a surprise, that we can't find any ligands or the classical ligands for L-selectin, which is PNAD and MADCAM, actually not detectable by conventional immunohistochemistry. And that, again, was a surprise to us. So we think there is a ligand for L-selectin in the tumor microenvironment, because we've shown that although those T cells, the L-selectin offers a benefit for activation in the tumor microenvironment, which I'm sort of not going to go into today. But the first thing we could show was that L-selectin was absolutely not required for homing to tumors, at least at the start of the therapy. And that was, you know, it was a clear-cut result, and this is where we started to work with Steve Paisley in the PET imaging. So here, what we've done is basically, this is the adoptive T cell transfer model I've told you about. So grow the tumors, sublethal irradiate, and then inject T cells on day 7. And what we've done here is we've injected 2 populations of T cells, ones that don't have any L-selectin and ones that do have L-selectin, side by side, and look at their recruitment into the tumor. So the green are knockouts and the red are L-selectin positive, and we could exploit our LDP T cells in this assay. So pre-transfer into the bloodstream, they're ratio 1:1. In the tumor, and the earliest time point we could do this was 18 hours. They're still 1:1. So both T cell populations are getting in there. So clearly L-selectin is not important at all. And in fact, the other surprising thing about this was that the spleen, we expected it not to be enriched because it's not an L-selectin dependent organ, but all of these other lymph nodes, either the tumor draining lymph node, which would be the sentinel lymph nodes, or on the other side of the mouse where we vaccinated, they also extract both L-selectin expressing and L-selectin knockout T cells. And this is not what you'd see in a naive mouse or a virus infected mouse. All these bars under the lymph nodes would be red. So we've got some very important remodeling going on in lymphoid tissue in tumor bearing mice that we haven't started to investigate yet. So as I said, that we now started to work with Steve, and this was a very productive collaboration. You've seen this image already. So just we exploited the fact that they had developed the zirconium labeling of T cells, and as Steve said, we put them into tumor-bearing mice, and we can detect them in the tumor. This is 5 days. What Steve didn't mention, and what is still a mystery, this is why we're working on it, is we see an awful lot of naive T cells in the liver. We see them in the spleen as well here, but we see them in the liver, and we don't know. And no one has really reported that before. So we don't think these cells are just dead. We try to extract them. We blocked them with antibodies to homing molecules, and it stops their location homing. So we've got to really work out what's going on there, and is the liver important in this therapy? But just as Steve said, these T cells are not affected by they don't lose therapeutic efficacy. So what we've got here is 2 tumors, well, 4 tumors, 2 that received zirconium labeled T cells and 2 that received unlabeled T cells. And you can see in all of them that once the T cells have gone in, they can all control tumor growth. This we thought this was validated the use of this. And then the other piece of information I want to give you is that with using PET-CT, we were able to track T cells injected in the tumor within 60 minutes. So this, as I said, it's naive T cells getting in there very early on. So this is a 1 hour time point here, and then this is radioactivity accumulating as the tumors grow, and this is the tumor growth by volume, a CT, and we also, it correlates with actual physical measurements. And it didn't matter whether you had higher L-selectin or not, it didn't really seem to have an effect. So just to finish, these studies, I think we've used different imaging modalities, and I think that's always going to be the key. I mean, I think trying to image exactly what's going on in a cancer at any one point in time is a moving feast. You're looking at basically it's a movie, and you need to be able to look at the whole tumor and everything that's going on inside the tumor, which is really, really quite difficult. But there are new techniques available that I think Arwin will may have spoken about already, but like the CADE mouse, where you can look at T cells that are infiltrating and then how long they stay there and when they leave. But anyway, I think that this is a point for discussion. So, but for us, what we think is we've revealed a way of manipulating L-selectin on T cells that potentially may have clinical benefit. So we're looking at whether we can do this in the clinical setting. So we've got Wellcome Trust ISSF funding to check this by using genetic modification. And we're also looking for non-genetic, pharmacological ways of maintaining L-selectin expression. We really want to ask whether L-selectin enhanced T cells benefit CAR T cell therapies, and that's a project that we're looking at. Emily Mills has just started that project in the lab. And really, we want to start asking, well, what is this link between L-selectin and clinical cancers? Is it to do with homing to, for example, of T cells to lymph nodes and better priming, or is it due to activation in the tumor microenvironment, which our studies have revealed, but I haven't really had time to talk to you about today. Okay, so I think the dogma would be L-selectin is boring, it's not on activated T cells, therefore don't study it. And we decided not to take that stance, but to study it. All right, and I just want to thank everyone. So, many wonderful collaborations in these are all the ones in Cardiff. And James Matthews, who really set up the melanoma model in the first instance with Arwin, and obviously continued collaboration and help from Arwin. So thank you very much. Thanks.
Thanks, Anne. That was fantastic. Thanks very much. I'd like to open up for questions. While people are typing though, because I haven't seen anything pop up just yet. I thought I might start off with a question. So Steve, if you're there, I'll start with a question for you. I was just I mean, obviously everyone's going to be hugely impressed by what's been possible at Cardiff University, and how they can apply that to their research. But I guess one of the key questions for me, who hasn't yet delved into it yet, would be, how long does it actually take to develop your own tracer? I mean, I know, like, obviously it's not going to be a straightforward feat, and there's lots of quality control checks that need to take place. So I guess I'm just curious about the sort of timeframes.
So this is a very good question. So PET imaging, potentially you could image anything at all, literally anything. It's because it's tracer specific. But of course, everything that you want to image, you have to develop a tracer for it. And so what I refer to as traditional PET, the sort of small organic molecule kind of side of things, there's a number of things that you need. I mean, if you're a biologist and you've got a pathway and you say, this is upregulated, I'd like to try and have a PET tracer to target that, and you're thinking of small organic molecules, but you don't even know the molecule, you're really in the realms of sort of AstraZeneca and Big Pharma to try and find what it hits that target reliably. And what a lot of the PET chemists do is they do go to clinical trials, or they try and look at things that potentially could have been therapeutics but didn't make it, if you see what I mean, because you give such tiny doses, something might not be suitable as a therapeutic but is suitable as a tracer, because you only have to give it once. You don't have to worry about build up and that sort of thing. But with organic chemistry like that, it is so molecule dependent. Sometimes a really good a chemist will just look at a molecule and go, oh, that was easy, I could just stick something on the side there. And then another similar looking molecule to the uninitiated, which kind of go, goodness me, that's really tricky. You'd have to build it from completely the other way to stop this part falling apart. And that's why I'm focusing on the biologics themselves, because I short circuit a lot of that process, if you see what I mean.
Yeah.
And so using an antibody, it's the same process. And if you gave me a batch of antibody, I do need quite a bit of antibody. It's sort of, if I was given free rein, I'd say, could I have 500 micrograms of antibody? But if you gave me that within 3 days, I could turn that into a PET tracer for research imaging. And there are, in terms of going to the clinical trials, of course, then you've got all the regulations and all of the approvals. So antibody and radio-labeled Herceptin has been in clinical trials about 5 or 6 years ago in the Netherlands to look at whether or not patients would respond to Herceptin. And we have got some clinical trials that we're just being asked to start in the UK, in PETIC actually, whether or not we could image patients with radio-labeled antibodies. But I think that would be the first one in the UK. And we actually, just to be clear, we wouldn't synthesize that on site. We'd be sent the tracer and we'd just inject the patients. So those antibody things and the zirconium is making its way into humans for the first time in the UK. And there are people paving the way for cell labeling as well.
Thanks, Steve. I better move on because I'm aware of the time. Oliver, I can see you've got your hand up. If I click allow to talk, would you like to ask your question? Okay.
Yep, can you hear me?
Yeah, I can hear you.
Oh, terrific. Really enjoyed both talks. I have a question to Stephen on the sensitivity in relation to the zirconium labeling of T cells or of effector cells. So if you label CAR T cells with zirconium, you inject them and they proliferate over a week or so. The radio tracer will probably be divided with each and be lower per cell with each division.
Absolutely, yeah.
And if you then have, let's say, not a bulky disease, but you have something disseminated, like peritoneal cells, malignant cells floating around, or leukemia, which is disseminated all over the blood and bone marrow, or we've discussed this, the meningeal space.
Yeah.
How quickly do you use the sensitivity? And then a second question, how long do you have to isolate a patient, or would you have to isolate a patient after you've used a zirconium tracer labeled product?
So these are two brilliant questions, and you've hit the nail absolutely on the head. So as the cells proliferate, there's just a sort of statistical sharing of the label between the cells, and it's not even exactly 50-50 with each division. It's just sort of roughly divided between the cells as they proliferate. Now, as you say, if it was a bulky tumor, the scanner really, to be honest, wouldn't tell the difference between, you know, twice as much activity in one cell or half in two cells, because it just doesn't have that resolution. But the dissemination is absolutely key. You know, if it starts to spread around the body, you start to dilute that signal out. And essentially, if it was just in blood pool that it was disseminating, what you'd effectively see is the sort of background signal of the whole mouse slowly increasing, or the patient, as you're probably thinking, increasing as the trace is just distributed around the body. In terms of numbers of cells and, you know, time scale, we could do calculations to work that out quite precisely, but I don't know off the top of my head, if you see what I mean. Mm-hmm. But in terms of the half-life of the tracer, as you saw from Anne's images, or some of my images, from the situation of when the cells stay put, you can see the image for 15 days. And then we've done some work to show just how, so labeling each different cell type, sort of, they have different efficiencies, because some take to the labeling better than others. But you can work out precisely exactly how many cells you can see at the start. And we could easily do some calculations before we embarked on a big study to see, you know, how quickly you think it would be dispersed, and that would be partly fed into the proliferation rate of the cells that you have. In terms of the other question about the patients, so one of the things, like, it's a very easy thing to say, and it's very often mentioned, and it's true, it shouldn't be forgotten about. You have radioactive patients if you're going to use this in the clinic. So what do you do with them? And one of the things is that we try and reduce the dose that we inject in the first place. So the fluorine patients who come in get an injection of a short-lived tracer, but they get a high activity and they get sent home that day. And the following day, the radioactivity is kind of completely dissipated. For zirconium, you wouldn't dare inject that much activity because you'd make them radioactive for such a long time. But it is worth mentioning that the iodine radiotherapy, so iodine's got a similar sort of half-life to zirconium, and iodine radiotherapy is a clinically approved process, and patients are given iodine and are sent home to get on with their lives. And you've heard the advice where you should just avoid contact with children and not share a bed with your partner and that sort of thing. So it's something that's not insurmountable, but it's definitely something that we have to sort of be realistic about.
Okay, great. Thanks. Super program, in any case.
Thanks then. Arwen, I think you had a question. Do you want to unmute and ask your question? I don't think it went up in the general chat though. If you...
Yes, can you hear me now, Helen?
Yeah.
Okay, I'm on a different computer to normal, so I'm trying to work out how it works as we go along. So my question is a bit similar, but it's for Anne, and I just wondered if you could tell us a bit about how sensitive PET is for tracking T cells. So what sort of numbers do you need in the mouse models? I know I should know this, but I can't remember. And do you think it's sensitive enough, Anne, to be able to tell you if T cells get right into the tumours, or if they're just hanging out and accumulating sort of around the edges? So this is more of a solid tumour question.
Yeah. So for the numbers, in order to detect we certainly had to put in more T cells than we needed for the therapy. And we used the PET in the end as a research to address a research question, and that is, were naive T cells getting into the, associating with the tumors very early on in the therapy. So for that, we did use many more T cells. As I showed you, they were still efficacious. And in fact, you've got really good tumor regression simply because we put in so many more T cells. With respect to location in the tumor microenvironment, or, inside the tumor microenvironment or not, I think that's a tricky one. And I guess it all, again, I mean, I'm not I think we need to refer to clinical radiologists. It's how that is done at the moment, and how that's decided on at the moment. I mean, I guess you could potentially, I think that's a tricky one. Yeah, I think that's a tricky one.
Can you take the tumours
I
out and look by flow or look by
Oh.
microscopy, or are they then radio-labeled and you can't work with them?
Oh, no, you could, no, absolutely not. Sorry, I thought you meant in situ. No, of course you could.
I meant both, actually.
Yeah, you could. I think it would be difficult in situ. I think what we saw in the mouse model is, obviously, as I said, we addressed this research question about, do the T cells get to the tumors very early on, and we got signals surprisingly within an hour. That's not what we expected at all, and that signal didn't wash away. Whereas in the lungs, I didn't show you, but in the lungs you see a very transient accumulation of signal over the first 4 hours, and then it's washed out. So we would say that signal is quite stable, stably associated with the tumor, and then of course we got increasing signal over days afterwards. And as you can't create radioactivity, that must be more cells coming in, even though they may well have have undergone significant proliferation, as Oliver suggested, and diluted out their label. We're still seeing accumulation of label. So you could collect the tumors and fix them, and subject them to IHC. You may be able to do something to detect label, like autoradiography. Steve will be able to address that. But with respect to flow, then if Steve had a flow cytometer in PETIC,
Right.
that would be a breeze, and that would be fantastic. We did try with that accumulation in the liver. We did do our best to try and extract T cells from the liver and see whether oh, there you go. Thank you, Rachel. It was still associated with T cells, but we didn't do it optimally. And but yeah, that can be done, and that's I think that's in the future research. Steve, is it?
Yeah. So, well, I mean, I do keep on applying for funding. So what I'd dearly love to do is, since I've sort of steered the research of PETIC along the lines of these sort of biological products, I would really like to be able to get some proper biological assessment equipment in PETIC, so that we exactly do those sorts of really exciting and interesting studies. I haven't been funded yet. I keep asking for the money. Oh, I keep trying. But to answer your question about the sort of microenvironment, I don't know if this answers your question or not, but one of the things we see very clearly with just the radioactive FDG, the glucose, in 41 tumors, is you get a big tumor with a hollow core. So you can, there's enough resolution in the PET that you can see a ring on the outer edge of the tumor, which is presumably the active outer edge of the tumor, and the center never takes up the activity. So you do get within tumor signal, but I don't know if you meant sort of at a more microscopic level than that, or as a bulk signal.
Yeah, no, I meant in general, whether they, I suppose this problem of them sort of hanging out around the edges is what I was
so
worrying about.
So for a completely different reason, with the FDG and just the way that the 41 tumors grow, they're very avid on the outside.
Yeah.
And you can quite clearly see a central part, and you can, analyze the activity take up of the different regions of the tumor.
Great, thank you very much. Can definitely see the value of having a flow cytometer in there.
Thank you, Arwen.
And also, I can see that Toby's asking a question, but it's been answered. Is it answered in the chat? Toby was asking if you can detect
in
circulating tumor cells.
I think, Toby, I think that, well, Steve can also answer. I think the real problem with that is you'd then be looking in the bloodstream, and then you've got the issue of any radioactivity that happens to be if it leaches out from cells, then how do you distinguish between cell associated and non-cell associated? But of course, if you could isolate those isolated blood cells and spin them down and separate them from plasma, and you can put them then in a nice little pot that Steve's got in PESIC and measure radioactivity and then phenotype them in some way. Yeah.
It'd be fantastic, actually, wouldn't it? Even for some of the preclinical models that Toby and I are setting up, I think that would be really interesting.
Okay, well, yeah, we can talk.
Yes, there's a comment in the chat from Paul Shaw. It would be interesting to irradiate tumours and see the effect on T cell trafficking, but not sure if this has been done yet.
Yes, it would be. You predict that you're stimulating a local inflammation, and I mean, things like STING, for example, STING agonists, so interferon alpha pathway, has certainly been shown to secrete the sorts of molecules that normalize blood vessels and even promote this HEV neogenesis. But I don't know if anyone's actually looked at it per se. But
there's a group in Melbourne that did pre and post brachytherapy tissue collections. I was actually talking about this to Arwen yesterday, and they did multiplex analysis of the tumor immune microenvironment and did see some changes in T cell.
Right.
Not live trafficking or anything, and it's sort of snapshots in time, but it did indicate that there might be some changes.
Yeah. Well, that's a good start point, because as I said, you need multiple modes of imaging to build the picture. Yeah.
Okay, I think that might be it for questions, but I did have one last one, actually, if I may, Anne. I just wanted to ask you a little bit about the F5B6 tumor model. When you added the 15C4 antibody for an efficacious response that correlated with an increased infiltration of T cells, did you also look at any other immune cells or other alterations in the tumor immune microenvironment, or just in the tumor microenvironment in general? Because I know you mentioned hypoxia and other things that might be relevant, but I guess what I'm getting at is there a difference between anti-VEGF type treatments, the large one, anti-large one treatments, and, you know, is there a benefit to one over the other, or beneficial for a certain patient?
Yeah, well, this, not surprisingly, is what the reviewers insisted on. This is, you know, really, Helen, I can answer, but this is John Greenwood's project in London, so I can certainly put you in touch with John. What his take on it is that this pathway is really a pathogenic pathway as opposed to a developmental pathway. So then it would be perhaps more advantageous to target a pathological pathway as opposed to a developmental pathway where anti-VEGF could interfere with, you know, other normal processes. So I think that's the idea. Yeah. What is striking, though, is that, because the reviewers did ask, does anti-large 1 and the combination of anti-PD1, does that induce HEV? Because these high endothelial venules, because that has been shown dominantly by, you know, Ruth Gantz and Gabrielle Burgess. Well, Gabrielle Burgess really has shown that. But in this large model, you do not see HEV at all. So, you know, so it's independent, and yeah.
That's interesting, isn't it?
Yeah.
I can see there's just one more comment in the chat that I will read out. Oh, actually, Toby, do you want me, if I unmute you, you can ask yourself. Here we go. There you go. Would you like to ask a question?
Yeah, thanks, Helen. So can only autologous transplant models be used? So, I mean, if you did it in a nude mouse, like a normal traditional xenograft, I'm guessing you can't do it because you need all the immune cells. Is that right? I'm just trying to plan what we can and can't do.
Right. No, it's a very, very good question, Toby. And hello, nice to meet you.
I am.
So no, I mean, if you think about, and Oliver will know this, Oliver's leading the CAR T-cell fitness program in Cardiff that I mentioned, that a lot of the CAR T, certainly CD19 CAR T-cell therapies were tested in immunodeficient mice first. So essentially where you take the patient's T-cells, engineer them and infuse them back into completely immunodeficient mice bearing CD19 expressing lymphomas. So you can do that. The problem there is if you're relying on multiple stromal interactions, and if the stromal interaction is between human and mouse, it may not be efficient if it was a mouse-mouse stromal interaction. So it really depends on your question, but we're planning to do it as part of our asking whether L-selectin enhancement works with CAR T cells. So we're planning to set those studies up.
Right, yeah, because we were always getting asked to use humanized mice, and then if I talk to people like Arwin and Alan Melcher, they're like, no, don't use them. You need to make mice, they're not very robust, etc. And I don't know. And now this could be really interesting. Yeah, you can answer some questions.
I think it's up to the regulators, because it's what they request before therapies can be tested in patients. And I think they are changing their mind, because Arwin and Alan are absolutely right. An immunodeficient mouse is just like a test tube in a sense, with human tumor and human T cells. So, and the stroke,
furry test tubes, Alan calls them, doesn't he?
Yeah. So it depends. I guess it depends what your question is. I mean, we would really question it for all the pathways we know are important for homing, because they just may not work. Human T cells may not recognize mouse ligands.
Yeah. Thanks, Anne.
Brilliant. Thanks so much, Anne and Steve, for joining us today. It's really instigated some really fabulous discussions around the field. So thank you so much for that. We've gone slightly over time, so thanks to all the attendees as well, and to Steve and Anne for hanging on to take all those extra questions. That was really fantastic as well. I just want to move on and just advertise the next cancer theme webinar. It's going to be on the 22nd of October, and Steve Napper is hosting Professor Alex Tonks from the Division of Cancer and Genetics, and he's going to be presenting about transcriptomics and proteomics in AML. So looking forward to that one next month. Thanks again for everyone for attending, and hope to see you then. Thank you. Bye.
Thank you, Helen. Yeah. Bye.
Bye.
Bye.